All right, this is a review for the units like nervous system and the immune system, reproductive system. And so we'll assume that we understand the basic idea of cell signaling, which is reception, transduction, response. Whereas you have a cell, and this is like the outside of the cell, and you have some sort of maybe this is a protein receptor, and then you have some sort of signal molecule, and the signal molecules are called ligands, and that binds with that receptor. Well, that's called reception. It causes some sort of response, usually another protein, some oftentimes called like a G protein receptor, something like that, which causes a shape change in that G protein, which will then activate another protein, and so forth. And this part is called transduction, where the signal is actually being transduced. And then in the middle of the cell, you have of course, the nucleus and the DNA. And the response is typically where this protein comes here and causes the production or the transcription translation of the DNA, which will then in turn produce, some sort of little protein which will go and do whatever this signal told it to do. And so that's basically how the signal transduction pathway works. Well, that has something to do with nervous, with immune, and with reproduction. Let's begin talking about the nervous system real quick. The nervous system is made up of nerve cells, and these nerve cells are called neurons. And a nerve cell kind of looks something like this. And then you have this tube that goes out here, and you have lots of little sprigs that shoot off of there. And you have, in the middle here, you have the cell body, and then this is the nucleus I'm drawing in red. So you have the cell body and the nucleus of the cell. And these little shoots that are coming off of the cell body are called dendrites, and the dendrites receive information from the surrounding nerves or from other signals. And this is the axon, is the sending part of the nerve cell. And then here you have these little sprigs that are kind of coming off here, and at the end of each one of them is the axon terminal. Now, also covering the nerve is this material called the myelin sheath, and the myelin sheath basically insulates the signal as well as protecting the surrounding tissues from the electricity, because the nerves use a electrical signal in order to transmit messages from the brain or from the body to the brain. Now, how does that work? Well, let's zoom in on the nerve. So here's your average nerve. And on a nerve, there are different gated channels. There are voltage gated channels. There are chemical gated channels, and these channels open and close based on the kind of signal that they're receiving. We have some voltage gated channels on the nerve. I'm just going to deal with the top side, but you get the idea. And this is the outside of the nerve, inside of the nerve. And also on this nerve, you have something called sodium-potassium pumps. And these sodium-potassium pumps pump sodium and potassium both ways. Sodium, it pumps sodium outside, and potassium inside. Now, when the cell is at this phase, you have what's called resting potential. The the cell is at rest. It has the potential to do something, though, and the outside is generally more positive, whereas the inside is a little bit more negative. And you look at that and you see the potassium's in there, and you think, why is it negative? Well, it has to do with other molecules that are inside the cell that aren't able to pass across the membrane, and proteins as well that make it more negative. And so what you would say is that there's an electrochemical gradient there. We've talked about concentration gradients before. This is an electrochemical gradient. There is the ability to do work. There's a charge gradient. There's a positive, negative. If you put two positives next to one another, they repel, but positive and negative attract one another. Well, they're attracted to one another, but there isn't anything that's quite set them off yet, so they're at resting potential. This is what we call this, resting potential. Now, at resting potential, the nerve can be acted on by something from the outside, typically an action potential from the rest of the cell, which we'll talk about what an action potential is in a second. Or it could be acted upon by some kind of chemical messenger, which we'll also get into. Whatever the case is, it causes those voltage gated channels to open. And when those voltage gated channels open up, sodium is going to go across the membrane because it is going to just simply diffuse from an area of high concentration to an area of low concentration. The membrane is no longer at resting potential, and there are no longer poles because the inside of the cell becomes positive. So what happened to the poles? Well, they were depolarized. So they were destroyed. The poles were destroyed. Now, quickly, sometimes you'll see a graph that has to do with this, and you'll see that the charge on the inside of the cell starts off negative, some number. Number's not that important. And then up here you have what's called the threshold potential. Now the threshold potential is where the charge needs to get to on the inside of the cell in order to fire the nerve impulse. So it's kind of like you need to have so much energy before it will go. It will either get enough or it won't do anything. So it's an all or nothing event. And so this is negative down here, this is positive up here. And so as the sodium floods into the cell, what happens is, this different color, what happens is the charge goes up. Now if it just gets to right here, nothing's going to happen, all right? But if it gets to that threshold potential, what ends up happening is it will go ahead and spike. And that spike is called the action potential. And another word for the action potential is just called a nerve impulse. Switch those two letters around. All right. So the action potential, well, it goes up, and this is when the cell is at its most polarized. It's the most charge on the inside. So the action potential is fired. That nerve impulse will then travel down the length of the nerve, much like a wave travels through a stadium. You can see it coming, it gets to you, you stand up, you cheer, do your thing, right? And so this is like a wave going through the stadium. Well, it gets to you, you stand up. You're doing the action potential. Well, what happens? You need to sit back down. And so the nerve needs to get back to resting potential. So what happens in order for that to happen? Well, the first thing that will happen is the sodium or the potassium inside the cell will now diffuse out of the cell. And what's that going to do? Well, that is going to repolarize or repolarization to the cell. And so what was positive is now negative again. And so what do you expect to happen here? That charge begins getting back to where it was to begin with. And what will happen it will actually over polarize, hyper polarize, and it's what's called an overshoot. And it'll send too much potassium out of the cell. And it does that in order to reestablish the resting potential. Now, how are we going to get back to resting potential? Well, we have to pump the sodium out and the potassium in. Well, in order to do that, we're going to use these sodium potassium pumps, and that requires energy. Active transport sets those back right to where they were, and the cell is back at resting potential, ready to fire a new action potential. Now, this period of time where it cannot make a new nerve impulse is called the refractory period. And so again, just to zoom out a little bit, if this is your nerve, the action potential is just going to be traveling down this, and the ions are going to be going in and out, in and out, in and out, and the action potential travels its way down the nerve. Well, that is called propagation. All right, propagation. Well, I told you about the myelin sheath earlier. So you have this nerve here. All right, and on this nerve, it is covered with these sections of myelin sheath, which are extremely nonpolar. Well, the action potential will actually jump over those, causing the conduction to be a lot faster than it would be without those. And so that is called saltatory conduction, where it's actually hopping over. Now, what happens when it gets to the end? So back here, I drew a picture, and the action potential can go down this axon and gets to the terminal. Well, it has to go to the next nerve, or it has to go to something else. Well, what happens when it does that? The nerves actually cannot touch one another. And if they do, they'll shock one another. So here's kind of a picture. This is a axon, and this is a dendrite of one cell. And in the middle, you have this space called the synapse. And this is the space between the two nerves. This is the axon terminal here. Well, inside the axon terminal are these tiny little sacs called vesicles. And if you remember from the cell unit, vesicles are just these sacs that contain some sort of material. Maybe it's going to be digested, maybe it's going to be shipped out of the cell. Well, in this case, it's going to be shipped out of the cell, and the material that is contained in these vesicles is called neurotransmitter. All right. So the action potential is traveling down this nerve, and once it gets to the axon terminal, you have voltage-gated channels here on the side of the nerve called calcium-gated channels. So instead of sodium rushing in, potassium going out, we have calcium that goes in. So what happens when the calcium goes in? Well, the calcium, all of it, will actually push these vesicles to the edge of the membrane. And remember the process we talked about, exocytosis? Well, those vesicles will merge with the cell membrane and release their neurotransmitter into the synapse. And so what was an electrical signal is now a chemical signal. And so up here we're electrical, and here we are chemical. So on this other side, you have some of these voltage-gated channels. Now these aren't just any voltage-gated channels, but they will only unlock with a certain mechanism, and they actually need one of these neurotransmitters. And so let me zoom in a little bit here. Here's like one of those channels, kind of close up. And so they are normally kind of closed like this. Well, this neurotransmitter will bind with that gate and cause it to open right up. Well, what's going to happen when that gate opens up? All the sodium that is outside the cell is going to rush in, and that's going to cause depolarization, which is going to cause a new action potential to form. And that action potential is going to go down this nerve. And so we've switched from electrical to chemical back to electrical, and those two nerves never touched one another. And it all has to do with that neurotransmitter. Now, there are several neurotransmitters I mentioned there in your notes. Acetylcholine is one of the most common. This has to do with your muscles contracting between a nerve and a muscle, the neuromuscular junction. Epinephrine, norepinephrine, dopamine, serotonin, several of those there. You should look over them. One other thing that we talked about with the nervous system is sensory receptors and how you have different kinds of sensory receptors. You have mechanoreceptors, how these are located in places like your ear, and these have to do with the stretching of these tiny little hairs. And you have nociceptors or pain receptors, and they detect pain using something called substance P. You have thermoreceptors, and they detect heat energy. You have chemoreceptors, and you have electromagnetic receptors or photoreceptors. That was pretty lazy. All right. Now, each one of these receptors detects some sort of stimulus from the outside, mechanical. Again, the hairs actually bend. The nociceptors, chemical, substance P, thermo, heat, chemo, some sort of chemical. And these are located in your nose and mouth. Thermoreceptors are on your skin. Nociceptors are everywhere but your brain. And electromagnetic receptors or photoreceptors are located in your eyes. Now, what happens? Basically, the way that all of these work, now there's lots more detail that you guys probably remember. We talked about the ear and the eye and all those different things. All right. And so what this is, this represents the sensory receptor. And the sensory receptor is going to receive some sort of stimulus from the outside, whether it's sound, vibration, or light, or some sort of chemical. And what that causes, that receptor will then trigger an action potential in the nerve. All right, and that will travel down the length of your nerves until it gets to the brain. Well, your brain does, so this is sensory. Your brain does something called integration, which it thinks. It actually decides what to do with that information. What am I going to do with that information? Well, then your brain finds another pathway. Let's say that you touch a hot pan. Well, your hand sends signals, a couple of them, thermo signals. This pan is hot. Nociceptors. This hurts us, and the brain says, well, we should do something about it. We should move our hand. The hand is unable to move without the brain's direct input, but the brain is unable to know anything about what's going on with the hand without this sensory input. And so sensory input goes to the brain. The brain says, all right, move your hand. The brain sends a signal back to the hand and says, move it. Well, that attaches to a muscle. We'll just pretend that's a good muscle. all right, and causes that muscle to contract, causing you to move your hand, all right? And then your brain will send out other signals that begin healing your burn. And so there's all these other things that are going on as well that you don't think about. And so that's the way that pathway works. We talked about several other things associated with the nervous system, um, but those are the main ideas. Now for the immune system, quickly. All right, a few things with the immune system that you should be aware of. We talked a lot about two different kinds of defenses. We talked about non-specific defenses and specific defenses. Non-specific defenses, these are things that you come with, you're born with. These are innate immunities, things that you're born with, like your skin, your mucous membranes, your sweat, the normal bacteria that live in your body, your stomach acids, things that you're born with, your white blood cells that are actually able to go out and whip things on their own. Your body also has something called the inflammatory response. When something happens and your body is attacked or injured, there is a response called the inflammatory response. Your capillaries will dilate, your venules will constrict, and this basically traps blood in this location. And this is happened by something called histamines. Histamines are chemicals released by the cells that are injured. And these cells attract phagocytes, which are these giant white blood cells, and they just come by and eat everything. And then they go and fight the infection. And macrophages, which are these other great big cells that come by and eat things, they go and clean up all the mess, and and so that the swelling eventually goes down, and that sort of thing. Well, that's non-specific. It happens no matter what. It happens when something, no matter what hits you, whether it's you get stuck by a pin or you get cut by a piece of glass, or for those of you who have allergies, your body triggers an inflammatory response against pollen or bee sting or whatever. Uh, Pyrogens, these are proteins that are produced by your white blood cells that cause fever. Interferons, these are chemicals that directly affect virus interactions. Not specific virus, but just viruses in general. And then you have your specific defenses. And specific defenses are against a specific kind of pathogen. And what is a pathogen? A pathogen is anything that causes disease, some kind of disease causing agent. And with the specific defense, you have something called MHCs, or major histocompatibility complexes. Close enough. All right, and you have two classes of these major histocompatibility complexes, class 1 and class 2. Easy enough. All right, class 1 MHCs. These are all of your other cells besides white blood cells. And when one of your cells is infected by some sort of pathogen, they will hold out the antigen basically on the outside of the cell. And so let's say you have here's your cell, and here comes a pathogen ready to infect your cell. All right, well, on this pathogen are these little signal proteins called antigens. Well, your cell will deal with that protein, or deal with that pathogen the best it can anyway, and it will hold out these little proteins on its cell surface so that your white blood cells know exactly what they are dealing with. All right, it's similar to being, like, say you're attacked, and you take a picture of your attacker and you hold out that picture so that the cop can see The picture is the antigen. It's the picture of your attacker, but your hand actually holding it out there is that major or histocompatibility complex. So your white blood cells will then come up and study it. So here comes Mr. White blood cell, and he's ready to. That's supposed to be like a police hat, and he's supposed to come up and help. And he's going to look at that thing, and he's going to say, I know what to do now. And so he sees them. He has a serious face on, and he's going to go out and deal with those pathogens. Well, he finds them, and of course they're all over the place. And he's going to take one and just gobble it up. Well, he eats it, but leaves behind these antigens. And he's going to hold those antigens out in order to show other white blood cells, look what I have here in my round hand. This is what I've killed. You go kill other things just like it. And this is what a class 2 major histocompatibility complex is. All right, and so those are specific defenses. Now we're going to get into some, the specific pathway that your body uses when it does this specific defense. All right, so when you're a macrophage, this police officer here that's eating your little sad faces in your body, kills something, he releases a chemical called interleukin 1. All right, and interleukin 1 is a signal to another kind of cell. All right, so interleukin 1 is a signal to another kind of cell. Now this is so we're making sure we're using correct terms here. This is a macrophage who's just eating something, and he's showing it, and he's releasing interleukin 1. Now interleukin 1 causes another type of cell to come up and read all about it. And this type of cell is helper T cell here. Well, helper T cell doesn't look all that great, but he is able to help you in your time of need. And his specific job is he's going to look at that pathogen. He's going to look at those those antigens there, and he's going to then form a plan of attack against that specific pathogen. And so he releases a chemical called interleukin 2. Now, interleukin 2 is going to rally two types of troops. And so this guy's name, helper T cell, and he's a kind of white blood cell as well. All these are. Right, helper T cell finds two kinds of cells. They find B cell. Nothing too serious about B cell. And Mr. T cell, or killer T cell here. It's more like a pumpkin, and he's not very angry looking. All right, and so a couple things are going to happen here. So we have the B cells that are going to be initiated, and what B cells do is this is part of your humoral immunity. Humoral is just like means your fluid, and this is actually going to be in your blood. Now, what they're responsible for doing is they're going to make antibodies. So what is an antibody? Well, again, so back to our little antigen. Antigens might look something like this, because all proteins have a shape. Well, your body needs to be able to form something that can deal with that specific antigen, because if you have, well, let's just do a close-up here of our bad guy. He's got these little triangles on his face. All right. Now, how is your body going to deal with it? Well, it's going to create a special kind of protein in order to do that. It's called an antibody. Antibodies are shaped like this, like a Y. All right. And on the ends of the Y are these special kinds of proteins. And this is how it recognizes what it's going to fight against. Now, your body doesn't automatically figure this out. It has to learn by trial and error. It has to learn based on what it's seen and what your other cells have told it, and it eventually comes about a protein. This happens in your DNA. Your DNA can form lots of different kinds of proteins. And so what happens is these antibodies will actually stick to those sites. And what that causes it to do is it causes that antigen or that pathogen to become useless. And it's also a signal to the macrophage to come along and scoop that guy up and eat him. All right. And so your B cells create those antibodies. And what's neat about your B cells is some of your B cells will go out and that their job is to make those antibodies. Well, you have other B cells, their job is remember, to remember the fight of old. So they set up a monument. I don't know, a little too much there. But they're called memory B cells. And they remember the fight. And so if you ever come in contact with that again, they're going to be able to immediately respond, which is why your second response to a pathogen is much more quick and a little more successful as well. Well, these kinds of cells over here are called cytotoxic T cells or killer T cells. And killer T cells are part of your cell mediated immunity. What does that mean? Well, if you mediate something, you go between it. And so the cell mediated immunity is where the cell itself is actually going out and killing the pathogen. And so these killer T cells will actually go around and find your infected cells, will go out and find the pathogen, and will use a protein called perforin. And that perforin perforates all of the infected cells. So it's like little protein bullets, essentially, that go out and kill all of those kinds of cells. Now, again, same kind of thing. You have some of those cells that will go out actively into the fight in order to deal with those little smiley faces or frown faces. And then you have others that their job is to remember, and they are called memory T cells, of course. And next time you have this kind of infection, you will be able to fight against that effectively. Now, this is the way that vaccines work. A vaccine is basically where you're given contact with a particular pathogen, and your body forms a defense against it, and that pathogen is not active. It's unable to reproduce for whatever reason. And so that your body forms a defense against it without you having to deal with the side effects of the actual pathogen. Like, say, for instance, a flu virus. You get a flu shot, and when you actually come in contact with the flu, hopefully, your body says, okay, we've seen this before, let's go out and kill it. And your body does it, and you don't feel any of the effects, or at least you don't feel the, uh, the extreme of the effects that you might have felt otherwise. A few more things on immune system. Allergies is where you have an overactive immune system. This is where your body attacks things that aren't necessarily bad, like pollen, that sort of thing. And then autoimmune diseases. Auto means self. And so this is where your body actually attacks itself. A couple of examples of this are, well, go back to our nervous system here. There's the myelin sheath. Multiple sclerosis is where your body is actually actively eating off this myelin sheath, which causes a couple things. It causes your nerves to not work the way they should because everything's going to travel slowly, and it causes a tremendous amount of pain in your surrounding tissues as that electricity is shocking your muscles now, whereas before it was not. Rheumatoid arthritis is where your body actually eats its own connective tissues as an autoimmune response. And so there's several of those autoimmune responses. And that's it on the nervous system and on the immune system. I'll have another video on the reproductive system and organismal development.